As Carl Linnaeus said, herbarium is better than any form of illustration, and every botanist should make one. And to carry forward that, I, Himanshi Kaushik, your host of the day, would want to welcome you all in today's webinar on insights into the world of herbaria. And I hope that you all are doing fine, sound, and happy at wherever you're taking this webinar from. So before we move on and learn a lot more about the herbarium, botany, and the information that we're going to get today through the session, I would request Dr. Gurmeen Rakra, ma'am, Assistant Professor, Admission Department Coordinator, to please introduce us and all the participants to our experts. Ma'am, over to you.
Very good afternoon to all of you. It is my immense pleasure to welcome the two resource persons of the day, Dr. Neeta Raj Sharma and Dr. Shabnam. Dr. Neeta Raj Sharma is a Senior Dean and Head of the School of the Bioengineering and the Biosciences. She is having experience of more than 25 years in industry, academia, research, and administration. She has been really instrumental in getting a lot of fundings for the national and international projects. She is a visiting professor at Birmingham University, UK. To her credit, she is having a total of 80 papers, 40 patents, 11 projects, and 10 books from the Springer Group. The second resource person of the day is Dr. Shabnam, who has done her master's and PhD in botany from Punjab Agricultural University, Ludhiana. During her PhD, she studied about the antioxidants and osmoprotectants responsible for the amelioration of the heat stress in cereal crops like wheat. Before joining LPU last year, she was working as a PGT biology at KV Kapurthala for almost a year. And she also served as an officiating principal at KC Group of Institutions, Namashare, for a period of few months. At LPU, she is currently guiding 6 PhD students and 2 master students under her supervision. Now I request Dr. Neeta Raj Sharma to introduce about the topic, Insights into the World of Herbaria. Over to you, ma'am.
Thank you, Dr. Gurmeen, for the kind introduction. Good afternoon and welcome all in our ongoing series of webinars. Today we have brought very interesting information and along with the demonstration for those who are aspiring to pursue their studies in plant science. Have you ever heard about the dictionaries of the plant kingdom? Well, that is called herbarium. A herbarium is a collection of plant specimens that are usually dried and pressed, and these are arranged in a sequence of an accepted classification and available for the reference purpose or for any kind of scientific study on the plants for the researchers, those who are just drying, pressing, and preserving the plant materials. But there are well-organized steps to be taken care of while making the herbaria. Even certain tools and equipments are required to make the professional herbaria. In today's webinar, we will be giving you the deep insights on herbaria, the procedural, and the importance of preserving the plant materials. Also, you will know about the places which are known for their herbaria. You will be given a small demonstration about the process so that if you are planning your career in plant sciences, you could explore this area as well. We have with us the plant science expert, Dr. Shabnam, and she will take you to all the details of today's technical contents on herbaria. So let's enjoy the session and enhance the knowledge on this interesting area of plant kingdom dictionaries. Dr. Shabnam, over to you. Let's begin with the session.
Thank you so much, ma'am. A very good afternoon to all of you. So as ma'am already said this, let's get into the world of herbaria. Before going into the world of herbaria, I think we are already into the world of flora So let's get back or past. I'm talking about my past. So whenever we were using our books, like here you can see, and whenever we were studying, we can find this kind of plant. I think this is known as Vidya Padhai. So we usually keep that. So the simple principle behind this herbarium making is this, which means the preservation, the drying of the samples or the specimens which are used into the day-to-day life, as well as into the more and more research fields towards the botany or towards the plant science. Now, talking about what herbarium is basically. So let me share my screen and move it. Yeah. So talking about the herbarium, so basically the herbarium is the singular form of the plural herbaria. So herbaria is the collection of the plants that usually have dried very well as well as pressed, because if any one part of the plant, if it gets distorted while preserving, it is not going to give any idea about the particular specimen. So the pressed and preservation method is very important. And herbaria is basically a single sheet on which a plant specimen is going to be attached. And as well as after attaching each sheet with the plant specimen, it is very, very important to follow the accepted system of classification. Now, talking about the classification, basically in botany, we use natural classification system as well as artificial classification system. Nowadays, apart from the Bentham and Hooker system of the classification for the plants, we are going towards the APG4 classification system, that is the angiospermic phylogenetic group classification. So this is all about the definition of the herbaria. Now, what is the need of the herbaria? So the first need of the herbaria is that whatever the plants, whatever the specimen of the flora which are available around us, we should know about that. The second thing is that it is very important to build the scientific knowledge, which means in the science students, it is very important if you are seeing a rose plant, okay, being an art student, it is nice to say that this is the rose plant, but being a science student, you should know the scientific name of that plant, that is Rosa indica, whether it is the Indian rose or whether it is the other country's rose like Rosa chinensis, that is China rose. Okay. Also, the another aim of herbarium is to preserve the catalog of seeds and dried herbarium plants. Also, because seeds, we all know this is the next generation. Okay, and it provides the full-fledged plant with the help of the principle of totipotency also. Okay, now next, coming to the main and major important aim of herbarium preparation is to maintain plants on the edge of extinction. Now, what I am meaning of extinction, suppose this is the red rose which we can see today. Okay, but we are not going to find it even after decades or next decades or maybe after 30 years. Okay, so this is the main reason. If one plant is going to extinct, from where we are going to have the characteristics of the plants which are already available in our past. So this is the main important aim of the herbarium, and also as it is very important for the students to know about how to prepare a herbarium sheet. Now talking about the steps, okay. So the first step of making of the herbarium is the collection. The second step is very important, identification. Now here I have especially put these identification step because whenever you find any literature on these steps of making herbarium or herbarium making steps, you will not find this second step on the paper. However, it is very important because when you have collected a plant and you haven't identified that plant in the fresh form, it is going to be very difficult to identify that plant after drying. So therefore, the identification step is very, very important. The next step is very careful pressing as well as drying of the plant specimen so that it could not be able to get too much dry that it will just going to crush all the parts. Okay, so the pressing and drying should be very well maintained. After that, after on the sheet, or we call it as the mounting of the specimen on the sheet. Next step is after mounting, it is very important to give the idea or the description of the plant, whatever the plant is available on the sheet. So labeling is the next step. And at the last, we need our future generations to look into this, that what kind of the plants we have preserved. So next and the last step is the storage step. So let's discuss one by one. First, talking about the collection. So while going out to the collection, we can go out to the field areas. We can also collect the plant species from the garden areas. Before collecting that, it is very important to decide what we are going to collect, what area we are going to visit. Also, what are the aim? Either we are going to collect all the flower specimens, or we are going to collect all the specimens which are fruit giving, or we are going to collect only algae plants, we are going to collect only pteridophytes or bryophytes. So this is very important to decide whether we are going to collect which kind of the taxa of the plant. The next information before going to the field is that you should have a proper literature, that if I am going to collect red rose, then which area is the suitable area for the collection of red roses. Okay. Also, for example, if I want to collect the pine cones, so the suitable area near to me is Himachal, which is near to the Punjab. So I can go to the Punjab. So it is very important that you should have a knowledge about the map as well as the different kind of geographical area. Next, and very important, if we are going individually, then it's okay. If you are going to have your teammates with you, so always carry a teammate, or always keep a teammate which is very, very motivated. Okay, he is going to help you during the adventure. So select that type of friends also, I can say, or that type of colleague while going out. And also one more important thing is the funding, because without money you won't be able to travel anywhere. Okay, so these are the main important decisions you should make before going out to the field. And next is carrying the equipment. So these are some of the equipments you should carry with you, like vasculum. So here you can see a vasculum is the box, okay, which is airtight box in which you can collect the specimen which is fresh in nature and put it in here, so that around up to 8 hours it is not going to wilt it. So this is the vasculum. Apart from the vasculum, you can use cutter, knife, tree pruners, polythene bags also, as well as field books, which is very, very important during the collection. Okay, as well as if I talk about your safety, so first aid box is very, very important, because while going out to the field, it is not very well known to you that which kind of the plants are having needles, which kind of plants are having spines in them, thorns in them. So you must carry a first aid box. Taking photographs is very important, because after, again, I'm telling you that after drying up the specimens, it is not going to easy to identify that plant, and therefore while collecting the plant specimen, you should take a photograph which will provide you with the idea about from which area you are taking the plant specimens. Okay, while observing, okay, or this is also known as the rough observation of the field, that what is the topography of the field, what is the type of habitat, whether it is a grassland, whether it is a coniferous forest, whether it is a rainforest, okay, whether this particular field is a desert area. So that observation should be noted on the notebook. Okay, this is one of the important description on which you can note different kinds of the note on the field notebook. For example, collection number, which type of number or which type of the plant you are collecting, whether it is 10th number, 4th number, 5th number, as per your list. Second thing is name of plant. It is not usually you are knowing the scientific name. But apart from the scientific name, you know the local name. If you don't know the local name, then just ask the local people what it calls. So note down the local name, or it is also known as the vernacular name. Okay. Province, which state you are going from, collecting the species from, which district you are collecting the species from, or which country you are collecting the species from. Also, nowadays we can use GPS system also to click the picture. So while clicking the picture, you can also give the idea about what is the altitude, what is the latitude where you are clicking the picture. Next is the habitat, its description, whether it is useful, whether it is poisonous. So that should be always taken care. And date of the collection as well as collector's name should also be mentioned in the field. So it is very important while collecting that how and what to collect. Do not over collect. Okay. So you can go to the areas which is very less known areas. So from there you can collect the plant specimens. Okay. You can also collect the plant specimens of your particular interest. Suppose you are interested in one particular flower. So just go and harvest the particular flower from different areas of the nation. Okay. Next, very important is that you should always must have learned that there are many plants which are common plants, and as we find them common, so therefore nobody is going to collect them. So try to collect common plants also. Okay, because common plants, they will provide you the idea about dominance of the species around that particular field areas. Okay, it will provide you what is the main feature of that plant which is going to be dominated at either desert area, either towards the grassland areas. Also, the last point is that whenever you are collecting the plant species, always try to collect the flowers if possible. Always try to collect the fruits if possible. For example, here you can see this is the flower of bougainvillea. So along with the twigs, along with the leaves, along with the branches, the flowers are also available with the plant specimens. Here, this is another example which is collected by our students of the LPU students. So this plant, in this plant, you can see very clearly the fruits are available. So this is very important to collect flowers as well as the fruit also. Now, talking about the collection of aquatic plants, it is very important to collect the aquatic plants. For example, Wolffia, as we all know, Wolffia is the smallest angiospermic flowering plant. Or to collect this plant, it is not possible to collect it one by one. So instead of collecting it one by one, you can collect a group of the Wolffia flower or Wolffia plants. Now talking about the Nilambo, the Indian flower plant, okay. So Nilambo, you can collect the thallus of the plant, one part of the plant, or either you can collect the flower of the plant. These aeroids are very difficult to collect because they have very lots of amount of moisture in them, and it is very difficult to dry them. So to drying them, you can also use a scooping method. Scooping method means you can scoop out the excess amount of the cells which are available inside the cell system. So after scooping out, just dry it and then paste it as it is shown in the figure. Collection of bamboo. So here you can see when there is a twig which is more lengthy. So in that length case, you can also paste that twig into the spiral manner, so that leaves are also shown, twigs are also shown, tender leaves are also there on the sheet. Collection of cactus is very important because succulent plants are more prone to the water concentration or water absorbing quality. So you can just go with the artificial heating system for the collection of these cactus. Talking about the collection of ferns, it is very important to collect the rhizome, that is the root part of the fern, because root provides a very distinct characteristics in the ferns. Okay, so this is very important that you should always include rhizome area along with the scales of the ferns. Okay, now talking about the palms. So palm trees which are very long in nature also, so you won't be able to collect the overall tree itself and just going to paste on the sheet. Instead of that, you can collect the upper side or the exile side of the leaves, which are arranged in a manner of the V shape. So collect that twig part and then paste it onto the sheet. Next and very important, this is what to collect. Now what not to collect. Okay, don't go to the areas for the collection which are prohibited by the government or by the local people also. So don't go towards that area. Also, if there are only few species or few samples are available, so try to collect only few samples. Do not destroy the plant dominance of the area. Next is, if you are going to do the collection, try to collect the functional part of the plant. That is, do not collect the sterile samples, and also do not collect repeatedly the similar kind of the specimen from different locations. The next step is the identification, which is very, very important. So for the identification, however, you have identified the common name, for the scientific name, you should always consult a specialist, whether it is your teacher, whether it is any kind of the taxonomist of the India or of the world also. You can send them the picture by mail, so they can also give you the correct naming of the scientific name. Okay. The another thing and very important thing is that nowadays, which is the technological world, we are saying that, and we are living in which we can also go on various online resources, like we have Systex. So this is a database system in which many different kinds of the plants as well as different kinds of their description is available. So you can match that description with the plant collected. Same as it is Tropicos, it is also one of the web site. Okay. Another one is the Natural Resources Conservation Services. Apart from that, you can also go to the Integrated Taxonomic Information System. So their link is also provided in this slide. Global Biodiversity Information Facility. So here you can also find the scientific name. Along with the scientific name, you are going to find the what are the numbers, approximate number of the plant species available globally. And how we will forget about this, our Indian Botanical Survey of India, BSI. So BSI many centers at India. So you can always go to and refer to the Botanical Survey of India website to identify the plant specimen. Third step is pressing and drying. Okay. So after collection of the plant species, so here you can see, you can use newspaper for the pressing and drying purposes. Okay. So in between the two newspaper, you should carefully keep your plant specimen, okay, without distorting the flower, without distorting the leaf. Okay. After that, after 2 or 3 days, you need to change this newspaper, because newspaper are going to take up all the moisture from the plant. Okay. And due to the moisture, it is very important that these plants are prone to any bacterial or fungal diseases or fungal attacks. So to prevent these attacks, you should, after 2 days, you should change the newspapers. Okay. Apart from these, using the newspaper, you can also use this kind of the presses, which is available on the right side, the end side of the slide. So these are the presses. After placing these plant specimens into the presses, you can just keep the press for around 1 week, and then after just opening it up, you can find the well dried specimen. Okay. So these are for the pressing. After pressing, it is also important that whatever the plant material you have collected should be devoid of any kind of the bacterial, fungal, or any kind of the infection. To avoid that, you should use mercury chloride or ethyl alcohol solution. You can also use formalin. Okay. So formalin is basically used for not changing the newspaper. So if we are using formalin, so there is no need to change the newspapers. Next, very important method or very important step is mounting or pasting the plant specimen on the sheet. Okay, so always remember that you should always place one specimen at one sheet. Okay, now talking about the standard size which is given by Indian government, as well as there are different governments which are providing the herbarium sheets. So the standard size of the herbarium sheet in India, we follow 29 into 42 centimeter. It is also followed by USA government. However, in case of the other government, that is in other countries like Europe, they use the standard size of 26 into 42 centimeters. Okay, now talking about the pasting, it is important you should paste the roots, tubers or rhizome at the lower side of the sheet, not on the upper side of the sheet. Okay, and after that you should use a straight line for the pasting. If your plant specimen is too long, then you can tilt at an angle so that it could fit the plant as well as it could fit on the sheet. Okay, so this is the picture showing how you can use the pasting. So while pasting, you can use glue for the pasting of leaves, tender leaves. And if the twigs are very woody in nature, which are not going to stick by the glue, you can use transparent sellotapes on that. That we will also see the demonstration. Next important step is herbarium labeling. Okay, so till herbarium pasting, it will be vain if you are not going to give the data which kind of plant is available. So first thing is that the size of the labeling, it should be 7 into 12 centimeter, and it should always be placed towards the right hand side of the sheet, which includes these particular particles. Okay, for example, name of the herbarium or organization. Let's say if we are talking about Lovely Professional University, so the name of the organization will be LPU. Okay, flora of districts or states, so whether we can write Fagwada or we can also write Punjab. Name of the family, for example, we have collected rose, so the family name is Rosaceae. Name of the genus and species, it is very important to follow the particular naming system given by the Linnaeus, that is binomial nomenclature. So you can write Rosa indica for Indian rose. Common name, as we know Indian rose as Gulab in India, so you can write Gulab also. Locality, altitude, frequency, that you can write by using GPS system. Okay, the date of collection, whatever the date you have collected, description or remarks or notes. So in here you can write any medicinal uses, it is used in medicinal purpose, it is used in any kind of cosmetic purpose, it is used in any kind of aromatherapy also nowadays very popular. And last is collector's name and number. Here number is depicting the roll number of our students. So talking about the naming system, okay, so Carl Linnaeus, as you all know, has given the binomial nomenclature of the species. Okay, so what is the main need of this binomial nomenclature? Let's discuss about that. For example, previously this particular plant specimen, which is known as the Ranunculus asiaticus, it was known as Ranunculus calicibus, retroflexus, pedunculus, falcatus, col, erecto, folis composites, which is a lot to learn. And the meaning of this plant name is buttercup with bent back sepals, curved flower stalks, erect stems, and compound leaves. So here you can find that how difficult it was to learn the scientific name at that time. And therefore, Linnaeus has given just two-word name or binomial nomenclature known as the bi, which means two. Nomial means name. So, which gives the genus as well as species name, and it, instead of this long scientific name, it becomes Ranunculus asiaticus. Okay, and that is the need by binomial nomenclature we are following. Apart from the binomial nomenclature, there are some rules also how you can write the scientific names. The first rule is that you should always italicize if you are typing on the computer, or if you are writing it by hand, we should always underline genus name as well as the species name separately. Genus name should always start with the capital letter. Species name should always start with the small letter. Apart from that, there are also some other rules. For example, let's discuss 2 or 3 rules only. Okay, so that you must have seen it is written Oryza sativa L dot or Oryza sativa Lin dot. The L word or the Lin word is basically depicting the scientist name. So here the Lin is for Linnaeus. Okay, so this is depicting the scientist name. Now talking about the other naming system, if there are 2 species system which is present, so you can also write the species by putting hyphen in that, and therefore we write Hibiscus rosa hyphen sinensis. Okay, so these are some of the important rules for naming. Now let's move to the last, storage purposes. So it is very important after preparing the herbarium sheet, you should always go to the storage, and these storage should be always put into the wooden or the steel cabinets. After putting these herbarium sheets into the steels and cabinets, it is always advised to spray fungicides for prevent them with, from the fungal effects, and you can also continuously spray the fungicides after 4 months or 6 months also. At the end, you can also arrange these stores as per the Bentham and Hooker system of the classification. So let's move to the next, that is herbaria which are present in the world. So okay, so these are some of the herbaria which are present into the world. So first one is the Herbaria laboratoria, okay, De Phenorogamia and Cryptogamia also, okay, which is available in France, and here there are around 100 lakh specimens of the plants are available. Okay, so let's quickly peek into their buildings also, how they are arranging. So this is the herbarium, okay, Museum National Histoire Naturelle Paris, and it is also one of the largest herbarium in the world. This is the Herbarium Royal Botanic Garden Kew, and it is the oldest herbarium of the world. Okay, so likewise there are other herbariums which are present in the world. So these are the herbarium which is available at St. Petersburg, okay, herbarium at New York, at Netherlands, London, okay, Botanical Garden Herbarium at Missouri. Now talking about the India, okay, so these are the important herbaria which are present in India, like Central National Herbarium. So here in the bracket you are seeing that it is abbreviated as CAL. So it is the universal abbreviation which is given to the each herbarium system, okay, and each herbarium institute. So like Forest Research Institute, it is abbreviated as DD, which is capital DD. It is situated at Dehradun, and okay, that is 3,30,000. So Central National Herbarium Howrah, this is also one of the oldest as well as the largest herbarium of our nation of India, okay, situated in Kolkata. It also serves as 2 kinds of the herbarium, monocot herbarium separately. They also have dicot herbarium separately. Apart from the monocot, dicot, they are now at the advanced level. They have digital herbarium system also. Okay, so these are very good places if you want to visit there. You can visit LPU also. We also have different kinds of herbarium present here. Apart from that, different centers of the Botanical Survey of India, Shillong, Coimbatore, Pune, they also organizes and they also preserve the herbariums. Indian Institute, Botanical Survey of India, Dehradun, Allahabad. Okay, Forest Research Institute, Dehradun, they also have one cabinet for the herbarium system. So these are some famous herbariums of our nature, of our nation. Now, last, role of herbarium. It is very important that why we are doing this whole work. So first role of herbaria is that this herbaria or these sheets, they act as the repository of dried plant specimens, safeguard them against loss and destroying by any fungal insects, and make them available for study. So the main purpose is that they are going to provide and they are going to aid for our future generation so that their study will be assisted. The next role of herbaria is that there are several herbaria which are available, and therefore they act as the proof of the existence of that particular plant specimen. Okay, and also if we are going to prepare that, somebody is going to identify that itself is going to create an original document. Okay. Another function of herbarium is that if we are collecting one particular or new species, okay, that should always be compared with particular family of the species or the same species of the same family, so that while going to the novel researches, we always compare our plant species with the past species, and for the comparison these herbaria plays important role. You can also use these preserved plants for biotechnological, for plant phytochemistry, for plant breeding and genetics also. Okay, and talking about the most herbaria, they also provide you many type of the knowledge. They can also provide you the information on the geographical distribution of the taxa, so where these particular species are going to present. Okay, so that's all from my side. So let's have a demonstration which is going to be given by Anas Hamishu. He is one of the PhD scholar of mine. So yeah.
My name is Anas Hamisu, actually, Mugby Professional University. I'm a PhD student under the supervision of Dr. Shabnam. I'm here to demonstrate the herbarium. Before we go into the demonstration area, we need to keep this into our mind that we need to have some facilities. For instance, we need to have a herbarium sheet. This is the Herbaria sheet. We need to have the Herbaria sheet. After the Herbaria sheet, we need to have a glue. After the glue, also, we need to have a scissors. A scissors in the essence that if we have the much leaves, we can reduce the size of the leaves. Also, we have the tank for helping us to stain the specimens that we're after. So now we are going into the real demonstrations of this herbarium. We have some plants species here. We have the palm leaves and we have rose leaves. So one of the important things that we need to keep into our mind in getting the herbarium, we need to maybe have the leaves, the flowers, the seed, and the stem, so that to help us to identify the particular species of plant that are about to become a distinction. That's the major importance of this herbarium. So now we're going into the the herbarium sections. Let's take for example, this is the dry rose specimen, and this is dry palm specimen. So now, since they have already dried, we can take two different. We are going to make use of the fresh specimen so that to see how we are going to make it clear for future reference. This is the red, white rose, and this is the red rose. So we have two different species of this rose. So we are going to take one after the other to demonstrate how we are going to make it on the sheet of paper. So now, as you can see now, uh, this is how we're going to do it this way. If this specimen is too long, you can follow it to the essence that it will not take larger size of the sheet now.
So here you can see that we are using this white rose. Okay, we can also use this red rose. So here you can see the length of these two roses are different. Okay, and if we are going to place this rose, we can place it vertically. But if we are going to place this rose on the sheet, we need to tilt it. Okay, so that this should be available on the overall sheets. So that is important. Yes, Anas, you can paste them. So for the pasting, it is very important that you should always use the upper side of the leaf. As it is very evident, there are two different sides of leaf of a plant. So the leaf or the sides of the leaf which is more shiny is considered as the upper side. On the other hand, towards the lower side, you can see that midrib is more protruding out. Okay, so we can use this type of the side, which is the upper side. Also, there are a lot of leaves which are not easy to paste on that, and each leaf is showing similar kind of the characteristic. So we can also exclude these leaves. So this is the overall specimen. Now talking about the twigging, we can also use scissor. Okay, and then this is now ready for our specimen to be pasted on. So for the pasting, we can use glue stick for the leaves. Okay, to paste the leaves, we will use glue stick, and to paste this stem, which is more woody type, which is not going to be pasted by the glue stick, we will use the sellotape, which is transparent in nature. So Anas, you will paste this on the sheet. Okay, so Anas is going to prepare one herbarium sheet at his home. Yes, so first you need to put the glue on the lower side of the leaf. Okay, yes.
So now we are going to apply the glue on the lower sides of the leaf.
Okay, so he is going to apply the glue stick on the lower side. By the time he is applying, okay, let me show you different kinds of herbarium sheets which are already prepared by our students. So it takes time and very much concentration. So by the time you can see different kinds of the herbarium which are prepared by our students. So here you can see that these are upright, okay, which are pasted along with the flowers. The another type is if the leaves are very broad, so you can also use leaves, only single leaves. Okay. Now here that is the point, that if there is no flower availability, you can use the whole twig along with the leaf only. Okay. So this is the palmately compound leaf. So these are the palm leaves. Okay, different kinds of the palm, date palm, okay, as well as sand palm. So this is if there are compound leaves are available which are alternately arranged. So you can use a overall twig along with the different arrangement of the leaf. So we can see that we are seeing only single leaf as well as different arrangement of leaves also. So this is the main idea of preparation of herbarium, that you can find very knowledge and different kinds of knowledge, that which kind of the flowers, which kind of the leaves which are present and how they are arranged on the flower. Now here you can see this is the overall inflorescence but not the flower. So this is the difference, that single flower is also available on the sheet as well as the inflorescence, which is the arrangement of the different flowers. Okay. Okay, so here Anas has already pasted the leaves. After the pasting the leaves, you can paste the twig. Okay. Yes.
Okay, as you can see now, we have already tested the specimens. We are now using the masking tape in order to make it more sticky together.
So it is very important, do not use the last side because it is going to be shown. Okay, so use the transparent side. Okay. Take this. Yes. So we are using sellotape because this is the woody part. Taste it. So this is the woody part which is not going to be stick by the glue stick. Okay. Also we are going to use the sellotape to stick this part, which is the flower part, so that it could not be able to fall. Okay, so this is all for the paste. The main important procedure is labeling. Okay, so Anas, could you please write the labeling, what we call it as? Okay. So the first labeling point is scientific name of the plant. And what is the scientific name?
The scientific name of the plant is Rose indica.
Yes, Rosa indica. So we are going to write the plant scientific name Rosa indica, and as I already discussed that the scientific name should always underline separately by underlining the genus as well as the species name. Okay, the family of Rosa indica is Rosaceae, so we are going to put here. The second thing is common name, already discussed with you all that in India we call rose as gulab. So we can write gulab, or we can also write it as Indian rose. Another thing, from where it is collected. So basically we have collected it from our outside of our school, that is School of Bioengineering and Biosciences, inside the lovely professional university campus. So we are going to write, okay, lovely university, lovely professional university campus area. Okay, so the date of collection, as you all can see, this is the fresh. Okay, so today it was collected, that is 1905 2023. However, we are using this fresh plant for today's demonstration only, but for the preparation of herbarium sheet we always use the well dried and pasted well dried specimens. Talking about the description, we can write in the description that it has red color of the flower, it has the leaves which are having the margins which are spiny margins. Also we can write any different kind of use. For example, it is used in cosmetics, it is used in medicinal purposes also. Okay, and then at the last, collector's name. So Anas has collected it, so we can write Anas Hamis. So that is all for the demonstration of this particular sheet. So here we can see that this is the particular herbarium sheet. Okay, so thank you.
Thank you so much, ma'am. And thank you so much to Anas as well for showing us how can we make the herbarium. And thank you so much for giving this practical demonstration to our attendees, to our participants. And in future also, when they will be seeing this webinar, they will be knowing a lot more information, practical view as to how they can, particularly make things work in the field of botany, and to be very specific in herbarium. So thank you so much, Shabnam ma'am, for giving your insightful experience, your presentation, and valuable information. And thank you so much, Neeta ma'am, for joining in and giving a boost to us, to all the participants as well. So, ma'am, with your permission, with Shabnam ma'am's permission, we have like some questions from our attendees,
Yes,
pre-questions.
ma'am. That was my last slide, which I could not be able to share. So let me share this. So, yeah.
Okay, okay. So, ma'am, there is this one question came from Radha. So she
Yes.
writes this question: does it require to clean our collected plants in herbarium?
Yes, very good, Radha. It is really very well pointed out that it is always important to clean your plant specimen. So basically, when we are collecting the plant specimen which are aerial in nature, okay, which are present on the above ground, okay, which are not towards the just towards the ground. So those plant specimens are already clean. Okay, if we talk about it through the rain, but if we are collecting the specimen, it is always good to clean the specimen with the help of filter paper, with the help of any botman paper, and you can use muslin cloths also to clear the paper. Yes, ma'am.
Okay.
Thank you, Radha, and very good.
Thank you, Shabnam ma'am, for resolving the query of the student. Ma'am, we have another query from Rakesh, and he says, how can we decide if this species is new or is it a subspecies of any particular plant? Or
Yes. Yes, so that is also a very important question, that whenever we are going to collect, sometimes that the plant which is very common, sometimes it has different features. Okay, so that becomes it novel. But how we are going to justify it that it is a novel plant or not? So for that purpose, you can always use different plant identifying app, or you can go to the websites which I already showed you, that you can also go to the BSI website. If you are not finding any kind of the similarity of your plant collected, you can always contact BSI scientists by mailing them the pictures that, sir, I have already collected this plant species. Could you please give me the scientific name? So they will help you at any time.
okay, I hope Rakesh, you must have got the answer that you've written here. So, ma'am, we have last question with this, but not the least, of course. This
Yes.
question reads, how can we prepare the herbarium and prevent our species from sprouting? I believe the student asking for any plant that sprouts.
Yes. So basically, the principle of herbarium is to excess out the is to just dry out the excess water. Okay, and if water is not going to present, it is not going to build also. Okay, water is very important for sprouting. If water is not present in the dried specimen, it is not going to sprout out. As you can see easily on your daily life, that we use black gram at our home, which is not sprouting if we are not going to give water to them. So this is the basic principle, that whatever the plant specimen is there, if it is not in contact with water, if it is not imbibing water, it is not absorbing water, it is not going to sprout.
Okay, thank you, ma'am. Thank you so much for resolving queries of all the students, the participants. We were getting a lot of admission queries as well. But ma'am, before that, I would like to ask Neeta ma'am if she wants to add anything to the webinar, ma'am.
Yeah, first of all, thank you very much, Dr. Shabnam, for your deep insight into the herbarium and herbaria, both, and also the equipments, tools which are being used for making the appropriate herbarium. This is not only for the botany students, but biotechnology students. They should also know, because plant tissue culture is a very important specialization in botany as well as in biotechnology, wherein students are required to identify the plants. And further, there is a very interesting area, that is the conservation of medicinal plants and the endangered species, wherein the herbaria is very, very important. And I am very, very happy that Dr. Shabnam presented the topic in a way so that the students who make the herbaria in general, they are very casual. They just collect and press and keep it inside the book, and they think that yes, they have stored. Not, this is not the right way. So this is the area which needs exploration, because India is known for its diversity, biodiversity. So this is how you can maintain your biodiversity. So I'm very happy that in this webinar series, we are bringing the topics related to life sciences, but very important.
Yes.
Thank you very much. Gurmeen, thank you very much. And the coordinator, she's coordinating very nicely. Thanks a lot. If any other question regarding the admission or any query, I'm there.
Thank you, ma'am. First of all, for giving this again, like for boosting us again, for boosting Shabnam ma'am again. And thank you, Gurmeen ma'am, for coordinating, for introducing us to our experts. And ma'am, with this, I take yeah, I take the permission to show our participants the site and the admission process. Can I do that, ma'am?
Thank you, ma'am.
Yes, ma'am. Yes. Thank you, Shabna ma'am, and thank you, Neeta ma'am. Neeta ma'am, your presence is always inspiring for us. And Shabna ma'am, you have taken us down the memory lane of our childhood days. And you have explained a very simple technique in such an organized manner that now I think every student will be listening this webinar would be able to preserve the plant specimens in a better way. Thank you, Shabna ma'am, for such an insightful talk. Thank you, ma'am.
Okay, then with this, I would be showing to all the participants our site, this one shot resolution for all the admission queries that we were getting a lot before the webinar also. So all the resolution will be provided on this site only, that is lpu.in. You can see lpu.in. So you just have to click on, or you just have to search it on the your browser, right? So once you click on your lpu.in, this will be the interface that will be in front of you. You can see these are all the highlights of the university that has either happened or may be happening or may happen in the near future, but of course all related to the university. Now you can actually explore our site in here. Now since we're catering about admission queries now, so we simply click on admission here. So once we click on admissions here, we have all the segregated programs right here. You can see after 12th, after graduation, after postgrad, doctoral programs, 10th, international, online education, and many others as well. So basically you just need to click on this admission and check it out in which course you would want to go. Now let's say we're taking an example of a student who has done his 12th grade and wants an undergrad program, right? So you simply click on this undergrad and regular programs. So once you click on regular programs, there are a whole lot of disciplines that would be visible to you, and all the disciplines that LPU has to provide to a student will be visible. You can see in here engineering, computer science, agriculture sciences, forensic sciences, all the disciplines that you just tell, you take the name, and we have in here. So I will be just in a brief telling you all about like how you can take part or how you can apply for the course that you would want to. So because we've had a good amount of talk about the botany today, so why not take the botany? So once we click on the botany in here, you can see that all the courses that we provide under that particular discipline would be visible to you. You can see we give you BSc honors in here in botany, right? So once we click on that, we can see all the program etc. That is under for a particular program. Now let's say we want to check the duration of a program, right? So the duration of the program is going to be 3 years, that is 6 semesters. And on the first screen that you see after clicking the course, you will be finding a duration. Now, after that, the most important thing for a student to check is the eligibility, whether you are eligible for a particular course or not. And to check that, we have program details, and after that we have our eligibility, and that is you should have at least 60% aggregate marks in your 10+2 with English, physics, chemistry, biology, or biotechnology. They should have been your subject in your 12th grade, and along with that you need to have at least LPU NEST or CUET qualified. So, and there are other options as well, like the relaxation, where you can get the relaxation, and all these categories and the conditions are mentioned in here. You can check it out, right? So now I'll just briefly tell you about the highlight. So highlight option basically gives you all the placement, research, student achievement, and rankings that one has to that the student either has achieved, and the rankings that a particular university has for a particular course. You can see in here we are ranked 3rd in ATL ranking of institutions on innovative achievements 2021, right? And similarly, you can just explore all the rankings right here. Now, there is one very important and interesting thing to note, students, is the curriculum part, that our university do provide you curriculum beforehand. Like in 1st, 2nd, and 3rd year, you will be given your curriculum even before you take the part in the admission in the course or in the university. Now is the hot topic, that is the fee structure. You can see in here the fee of a particular course is 50,000 rupees per semester, this particular course. Now again, this is not the usual fee that we pay. Why? Because our students get a lot of scholarships. And how and from where they get a lot of scholarships, and these are the basis of those scholarships. You can see at least they should have a 95% in their 12th grade, or they should have at least 330 marks around from 400 in their LPNS test to avail this 28% program fee waiver on their per semester fee, right? So here basically our scholarship are divided into 3 slabs or 3 categories, 1, 2 and 3, and you can check in here what category your marks to fall in, and after that you can similarly apply for that scholarship. Right now, let's say you were not able to get any of the scholarship mentioned here. Don't be sad, because we have other scholarships as well. So we have scholarships on the basis of NEET. We have other scholarships on the basis of innovation, startup, or entrepreneur idea that you may be a part of, or are you a board topper? We have a scholarship for you, and there are other scholarships for the sports and cultural and co-curricular activities as well. And we have some financial aid scholarships as well. So whatever scholarship you fall in, start applying for the scholarship and start applying for the course. Now is the important date, and this is of course the most important part for the student right now, is the last date of the admission is 25th May 2023. So students, I would urge, it's not too far now. I would urge you to start applying for the course, start applying for the test, so that you can get your favorite course, you can get your favorite stream, if whichever you want to take part in. Now is the basic question is how you can apply, and here is this one thing, that is, you just simply click on this apply now option, and once you click on this apply now options, there are these bare minimum requirements that will be asked from you, like your name, your gender, your address, your state, and after that you click on submit, and you will be given your dashboard. And once you will be given your dashboard, you can simply apply from there. Like there will be whole options available to apply for the test and to apply for the course or admission. And I hope that I have given you all the resolution and the navigated you all the way how you can take admission and where you can find a particular thing for a particular course, right? Even if you have other queries that I could not address in this webinar, we have all the options available for you. So you can either schedule a call, you can either WhatsApp us, or have a live video counseling session as well. Now, last but not the least, these are the numbers. You can just simply write it down somewhere, or maybe take a screenshot of it. You can call on these numbers, and our counselors will be here to provide their assistance and to resolve your queries. And I hope that I have given you all the details as to how you can take admission in your particular course. So I would urge you, I would request you to start applying for the course, start applying for the tests, so that you can get your favorite course and test. And with this, I would ask permission from all attendees, and of course our experts, Neeta ma'am, Shabnam ma'am, and Gurmeen ma'am, to wrap this webinar up. So with your permission, can I?
Yes, ma'am.
Yes, ma'am.
Okay,
If there
then.
is no query further.
Yeah. Okay. I believe, ma'am, there's no query in here.
Okay.
So, yeah. So, I would say at last, thank you so much for joining in, ma'am. Thank you so much to all the experts in here for giving your time, for crediting your time, and giving your experience for all the attendees and for me also. And I would like to thank to all the attendees as well who's going to be seeing this webinar maybe later, for being patient with us, for being valuable for us. And with this, I take your leave, and until next webinar, stay safe and stay healthy, and thank you so much for joining in.
Thank you, Himanshi. Thank you so much for coordinating this evening.
It's all pleasures mine, ma'am.
Looking forward another interesting webinar.
Definitely, ma'am. Definitely.
Thank you.